Hello everybody and welcome to our video today where we are talking about biology chapter 12, evolution. So before we get into the details and the vocab of this chapter, it's important to make note of some of the experiments that were done to determine that DNA was in fact the genetic material. So first we have Griffith's experiment. He used mice, injected different strains into these mice and basically discovered that transformation. He discovered transformation. He put a virulent strain in a mouse, okay, and that mouse died. He put a non-virulent strain in the mouse did not die. Then he put a heat-killed virulent strain in the mouse stayed alive. However, when he took the non-virulent strain and the heat-killed virulent strain, that mouse actually died too. And so what he concluded from that was transformation. Next we have our Avery experiment, and they were just discovering that what happens if you degrade DNA and what happens if you degrade protein. So this is when they still were unsure if it was the DNA or the protein that was actually the genetic material, and through that they were able to determine that it was the DNA. And then we have Hershey and Chase's experiment, which used radio labeling of DNA, and they were also able to see how this DNA was passed on. So these you can look up to look in a little more detail, but just to give you the background information on all of them. So we have different types of evolution. So we have divergent evolution, which is when the species start at this in the same place, but then they diverge and they start evolving into separate organisms based on the different environments that they are in. Then we have parallel evolution, which kind of deals with species that have a more like a closer common ancestor compared to these two types of evolution. Then we have convergent evolution, where species start off as separate, but then they start convergent evolution, and they start evolving into having some similar characteristics based on the environment that they were both kind of placed in. So those are just some things to watch out for. On top of this, we have Mendel's laws about genetics. So he had a law of segregation, which stated that an organism has two alleles of each gene, and that those two alleles separate during anaphase 1, which therefore allows the gametes to carry only one of those alleles. So when we get into our biology, like cell reproduction, we'll kind of look at that in a little more detail, but that goes under Mendel's laws. And then he also had a law of independent assortment, stating that the inheritance of one allele does not influence the probability of inheriting a given allele for a different trait, except for linked genes. That, as we know, and as we'll talk about in another chapter, those two things can be inherited together. Certain genes can be inherited together. However, his law of independent assortment was just saying that for one allele that is, not a linked gene, by inheriting that allele, it has no influence on the probability of inheriting another allele for a different trait. So next, we have a little bit of vocab. This chapter is a little vocab heavy. So we have penetrance, which is the proportion of people that are carrying an allele that also exhibit that phenotype. Okay? Then we have expressivity, which is the varying phenotypic outcomes of a specific genotype. We have genetic leakage, which is when the flow of genes, when there is a flow of genes between species, and this leads to hybrid offspring. A common example of this would be like a zebra horse. You can look up a picture. So that is due to genetic leakage of a flow of genes between species. Then we have genetic drift, which is a change in the gene pool just due to chance. And we have the founder's effect, which is a bottleneck, which is again the change in a gene pool, but this is due to a specific event that took place. So that's bottleneck effect. So the founder effect is a bottleneck that isolates a small population and causes there to be interbreeding. Then we have the modern synthesis model, which just states that the mutations and recombination lead to mechanisms of variation, which we know. There is inclusive fitness. So if a population meets a certain criteria, the allele frequency will be constant. And we will look at this a little bit later when we talk about Hardy-Weinberg's principle and equation. So just remember this one. So if a population meets a certain criteria, their allele frequencies are going to stay constant throughout that population. Then we have punctuated equilibrium. So, that just expresses that evolution is a slow process, but there are bursts of evolutionary activities. So that's punctuated equilibrium. Then we have isolation, which is reproductively isolated by a pre or post zygotic mechanism. Then we have the molecular clock model. And this is the degree of difference between two species' genomes. And they're saying that that difference in their genomes is related to the amount of time since their common ancestor. So this one, I'll read it again. The molecular clock model is it's stating that the degree of difference, so the difference between two species' genomes, is related to the amount of time that has passed since they have evolved from a common ancestor. So that's how you can kind of tell and put things on an evolutionary timeline, is based on their genomes and how different or similar they are from certain other species. Then we have different types of selection. So we have stabilizing selection, which is when it's trying to keep a phenotype in a narrow range. So stabilizing selection, keeping a phenotype in a narrow range. We have directional selection, which moves the average phenotype towards an extreme. So directional is kind of pushing it towards one direction. Then we have disruptive selection, which moves the average towards two different phenotypes at extremes. And this can cause speciation. And then we have adaptive radiation, which is just the rapid emergence of multiple species from a common ancestor. So these are definitely some important words that you should just know. I've definitely seen these in some practice problems that I've done, especially down here, the stabilizing selection, directional selection, disruptive selection, and adaptive radiation. So I definitely recommend, knowing those. And then we have one more word here just to finish off this chapter. So we actually have a couple more concepts to cover. So for one, we, I kind of skipped over this kind of basic foundational vocab. So we still have alleles, genotype, phenotype, dominance. So those are all words that I'm assuming if you're watching this video, you already know them. If you don't, definitely look them up. So alleles, genotype, phenotype, recessive, dominant. Okay. So next, we're going to look at deleterious dominant alleles. And this is saying that they will remain in a gene pool if the organism's fitness remains unaffected, even if it reduces the survival of that organism after reproductive years. So a deleterious dominant allele, as long as it doesn't change the organism before its reproductive years, that allele is still going to be in the gene pool, because by the time you notice that this gene is or this allele is a problem, that organism will have already reproduced. So there's that is how this kind of allele will stay in a population. And then you have deleterious recessive alleles, and this allele will evade elimination by natural selection through phenotypic masking. So if you have a deleterious recessive allele, if it's heterozygous, the dominant trait is what is going to be expressed. So, that again will also evade elimination by natural selection. So, switching gears a little bit here, just to bacteria, there's, I think we go over this in an earlier chapter as well, but just to make sure we cover it, we have transduction, this is basically how bacteria receive genetic information. So we have transduction, which is the transfer of genetic information via a bacteriophage. We have conjugation, which is the transfer of genetic information via a conjugation bridge. I would look up a picture of this one as well. Then we have transformation, which is the bacteria that gets genetic information from their environment. So that is transformation. Next, we're going to look at chromosomal mutations. So we have deletion, duplication, inversion, insertion, and translocation. Most of those are self-explanatory, so I'm not really going to go into detail. But one important thing here is truncated proteins. I didn't know what that word was initially, so what that means is that it results from a nonsense or a frame shift mutation with a downstream stop codon in the new reading frame. So we know the stop codons are UAG, UGA, and UAA, but you will get a truncated protein in a result from a nonsense or a frame shift mutation that causes there to be a stop codon in the new reading frame. So that was something that I've, found out that is something I should definitely know. So I wanted to share that as well. But then we also have nucleotide mutations. So we have point mutations and frame mutations. So the point mutations is just when you will be switching one nucleotide for another. And then a frame shift, so we're reading the codons 3 at a time. If there's a frame shift, that will throw off all the other frames. And this will cause, both of these things can cause a silent mutation, which is, where it was replaced by a nucleotide that also codes for the amino acid that you're looking for. So in that case, it's a silent mutation. It's technically not supposed to be there, but it didn't really cause any harm because the correct amino acid will still be produced. Then there's a missense mutation, which is when the wrong amino acid is produced due to a change in nucleotides. Then there's a nonsense mutation. And we have insertion and deletions as well, which again change the reading frame. So, and the last part of this chapter focuses on analytic techniques and recombination frequency. So the recombination frequency is just saying that the farther apart two genes are, the more likely they are to recombinate. I'd say that's just, a background thing that you should know, that the farther apart two genes are, the more likely they are to go through recombination. And finally, here we have our analytic techniques. So we have our Punnett square, which again, if you're watching this video, I'm assuming you already know what that is. If you don't, please search it up. And then we also have the Hardy-Weinberg equation. And this is something that was new to me when I started. So I would definitely recommend looking at some examples. But the Hardy-Weinberg equation is based on the inclusive fitness that we said before, that if a population reaches a certain criteria, the allele frequency will remain constant. And so this equation is kind of the application of that. And so you're going to use P and Q. So P being the dominant, the dominant allele, Q being the recessive allele, and then PQ being the heterozygous, right? So the Hardy-Weinberg equation says P plus Q equals 1. So, and then P squared plus 2PQ plus Q squared equals 1. So with these two equations, you're able to tell, the frequency or how many of a certain, out of a certain number of the population, how many organisms are dominant or express the recessive allele, or how many are heterozygous. So this is just playing with these two equations in a few different examples. Like I said, I would definitely suggest looking that up right now. Just look up Hardy-Weinberg example. So yes, just know these two equations though. But that leads us to the end of our video for chapter 12. If you have any questions, please put them below and stay tuned for our next video.